the ethics. I heard a lot of great kind of debate and questions during the break, so I'm interested to see what happens in the second half. We're going to start with you up front. You had your hand up, so what was your question?
I just enjoyed saying, other than the complex stuff with epigenetic regulation, what other problems are there for eukaryotes?
So you're thinking about the problems with the complexity arises as we go to more complicated organisms. Before you answer, Chris, what do you think some of the complications would arise naturally from going to a more complicated organism?
I would say the biggest thing is if you have like bacteria. Well, let's say the simplest thing. Suppose you want to make, suppose you just want to make a chemical with bacteria. You just put the gene in and they make it, like the green fluorescent protein example that people do in high schools now. But if you're messing around with animals, you obviously don't want them to be making green fluorescent protein in every single cell, unless of course that is what you want, but it's probably not. Then what do you do?
Alright, so first of all, in terms of eukaryotes, it's not that we can't do eukaryotes at all. If you're doing single gene type stuff, that is no problem in eukaryotes. People do it in mice all the time to study mice biology. You call those transgenic mice, and those are, I mean, you're affecting the germline of those organisms so that the changes you put in get perpetuated. You can buy GFP zebrafish. Actually, you end up playing a different version of the talk than the one that had the picture of the zebrafish, but you actually can put GFP in every single cell of an animal and the thing still be totally happy. But it's fairly easy to restrict the expression of a gene to a particular tissue. That would be an example of regulation. That is, again, routinely done already. The main division, I think, when you look at the eukaryotic space is whether you're going to affect the germline of the organism or not. So there's nothing stopping us today from making like GFP tattoos. And that would be something that doesn't affect your germline, it's just something that's going to locally affect the skin cells on your arm, say. But still, it's a genetic change to those cells, but when you have kids, it wouldn't perpetuate because it's not in the germline. If you really were going to, genetically engineer human beings the same way we can manipulate a mouse, you would have to be able to do all the sort of embryonic stem cell and cloning sort of things that we do to mice with a human, which would definitely be unethical. I think in pretty much everybody's opinion. Not everybody's, but most people's.
We have a question over here against the wall.
So when trying to deal with tumors using the liposomes, how would you differentiate between malignant tumors and benign tumors, or would you want to?
Well, there's no real problem with why not affect a benign tumor? It's still a tumor. So I don't know that you'd necessarily try to avoid benign tumors in the process. It would probably actually be tricky to make something that was selective for a malignant tumor over a benign tumor. The only real differences are going to be the cancer-associated epitopes. There are some things that would not be present, like beta-1 integrin is usually not present in a benign tumor, but would be present in a malignant tumor. But why you'd want to achieve that, I'm not quite sure.
Also, how long would it take from injection to affect the malignant tumor?
So we envision this as being something fast. So, part of our process is invasion, and invasion itself is like a 30-minute process. Our bacteria, we engineer them to invade and then do this kind of payload delivery thing where the bacterium actually pops within the cell, delivering all its contents. And that is about a 5-hour process. And we intend this, we envision these things as being like a 24-hour type thing. What then happens after that is could be a much longer time scale. So in general, bacteria are always operating on a minutes to hours time scale, and everything eukaryotic is working on more like an hours to days or longer time scale. Just everything's a little slower in eukaryotes.
But how is that so different from what's done now? If I go get some chemotherapy right now, what are we talking about a wildly different time scale, or are we pretty close?
Well, I think you usually do those, I mean, that's not just a single dose. Radiation is usually a single dose or a few rounds of single doses. And in that sense, it's very similar to the kind of the timeline of what happens. But then you have slow responses to that. I mean, you don't do you don't do the radiation and instantly.
I'm not running around
regression. It takes a while for your body to respond. I mean, the cells are probably already dead, but then what happens to those cells?
So with these kinds of treatments, you would expect the body's recovery cycle to be better because it's more targeted. Maybe not. Okay.
Not necessarily. It would probably be on the same time scale, just because, I mean, what's going to happen after you kill a cancer cell is you're going to have, kind of the cleanup reactions, which are done by macrophages and them to clean it up, and you're not affecting that time scale.
Where was our next question? Right there.
You were talking earlier about people going down to Antarctica and getting a bacteria and getting some useful thing. You still have to do that to get the gene that you want to put into the E. coli, right? But the other question is, once you find that gene, whatever it does, how often do you have to modify the gene to make it more potent or more effective or more efficacious?
Those are both great questions. So the first part is, we don't necessarily have to discover all new gene functions because people have been doing it for 30 years, collecting gene functions for us and describing them in papers and putting their DNA sequences into databases. And since we can just synthesize things, we don't actually have to have the physical DNA from it. So if we want a gene from a mastodon, as long as someone has sequenced that mastodon and described the function of those genes, we can just synthesize those genes. So we don't have to have like a fresh biological sample of anything. We just need the information. The second question, though, is applications that require new parts. I'm rephrasing what you said, but in general, that's what you mean. And largely, this field is thus far focused on applications where the parts you would need to build the device are already described. So you just go into PubMed and search for the functions you want, and hopefully it already exists out there in nature, and you just make that DNA sequence. And a lot of things can be done that way. Most of these biofuel efforts are of that ilk. They don't necessarily have to make new enzymes to make the pathways to make the biofuels. They have to tweak them a lot to make them express at the levels they need and make them stable and so forth. But all the enzymes already existed somewhere out there in nature in some other context. But there are a whole lot of applications where it's just not something that nature has provided. And actually, that's one of the areas that we work on in the lab, is how you identify new parts that just are fundamentally unnatural, that are necessary to make your application. So, you can grab all the genes to do a biofuel pathway because carbon metabolism is just incredibly diverse out there in nature. So there's probably an enzyme out there that already does those reactions. But let's say you wanted to make perfluorocarbons. So perfluorocarbons are something we do chemically, but are actually very foreign to biology. You don't get much fluorine chemistry at all in biology. So if you wanted to, say, make industry scale volumes of those sort of chemicals using a fermentation process, you would have to make enzymes that did the individual chemical reactions.
We're going all the way in the back.
So one of the things you talked about is production of these things and the whole process that you're going through. You're trying to speed it up with these robots. How much of this is being simulated in software? And also, is the state-of-the-art inhibited by technique, like algorithms, or is it inhibited by computational power technology?
So
before you answer that, what's the motivation for that question?
So, well, in college I roomed with a few bioinformatics majors, cross between biotech and computer science. And it's an emerging field, and I'm just curious what it is in this context.
And when you saw all the information that Chris put up there in his presentation, did you initially think, hey, this is a great place?
Yeah, it kind of like hurt me emotionally to see all that stuff being done in real life instead of in software. Like that could be it could be simulated. Why aren't you doing that?
So so what could be automated in your mind? And that's the last one. I'll let you go after this. When you said,
well, I don't really understand a lot of this biology, but I mean, chemistry is pretty well understood, and if you could have inputs and outputs, and like, you know, what situations cause other situations, that's what software does. You could simulate that.
Yeah. So the major limitation right now, the reason that modeling and simulation is not pervasive yet, and I wouldn't say it's absent, I didn't talk about it so much. In some spaces, that's really common, and it's fairly diverse. Like, there are many different applications. Like, for example, in biofuels, if you go into these companies when they're trying to build the pathways, they definitely use bioinformatic type models and software tools to kind of identify the preferred genes to make into their systems. And if you're looking at the kind of dynamic circuits, it's very common to do differential equation type modeling of the dynamics of those systems prior to building the systems. The thing that is truly limiting, though, right now is the DNA synthesis aspect of it. So both in terms of the cost of making a given DNA and the time associated with building that DNA, that's 90% of what you end up doing. But that's all going to change very quickly. No, it's physical. Until it's automated, you could simulate it, but it wouldn't be interesting to simulate. I mean, it's all repetitive tasks. It's just repetitive tasks that thus far have not been made automatable. They require manual manipulation.
quick follow-up. How much does it cost to synthesize a DNA right now for some of the work you're doing?
So if you do things by what I term total synthesis, which is the gene synthesis technology where you're starting with the chemical process to make the oligos and then assembling them up, it costs, depending on how quickly you want it, between 50 cents and $1 per base. So you're saying then every single gene is about $1,000. Every single one of these devices is something like $5,000 to $10,000. And in academia, that is limiting. We actually cannot use that technology ubiquitously. We will use it, but we also use other more traditional cloning techniques that involve natural DNAs to kind of piece things together. In industry, they actually do both as well, but for them it's not the money, it's the fact that the time is really problematic. Gene synthesis right now is a fairly slow technology where for some DNA, like if you were to order from a company something that's 8,000 base pairs, you're probably looking at about a 2-month turnaround time.
Come on, Chris, it's always about the money. This is science.
It is for me, but I'm cheap.
Okay, a couple things. First of all, was the glow-in-the-dark bunny produced this way with the E. coli?
Glow-in-the-dark money?
The bunny, you know,
bunny,
rabbit.
yes, absolutely.
Okay.
Yeah.
What makes the E. coli a special engine that isn't possible with other kinds of bacteria?
So there are two reasons that everyone gravitates towards E. coli. One, it is incredibly manipulable. It is much easier to change the DNA of E. coli, and there are more tools to do it than any other organism out there, such that every cloning process out there, even if you're doing bunnies, you're first putting the DNAs and building them in E. coli and then moving them to the bunny. The second issue is information. It's just by far the best characterized organism. We know what pretty much all the relevant genes are within it and what they do, and there's just tons of literature about it. Because we had this long period from the 70s through now where people were just cloning genes. You get more information about heterogeneous transfer of genes from other organisms to E. coli than from any other permutation of one organism to another. And when you go to design something, effectively what you want to first of all know is what's going to happen when you put this DNA into the cell. And you don't have that information for other organisms, so you'd have to do the basic science first.
And it's simple and it's cheap too.
Relatively speaking, it's cheap and it's fast. I mean, one of the main limitations to doing eukaryotes, like a human cell only divides once every 24 hours or so. And if you're trying to do cloning, like where you have to isolate genetically pure things, which is something fundamental to doing genetic manipulation, that you have to do that. You know, you might have to wait a month or something before you get a clonal population of a mammalian cell for certain cell types. But E. coli, they divide about every 20 to 40 minutes, so a 24-hour cycle time gives you a clonal population.
I have a question right there.
So after going through the treatment, what are the chances of the cancer coming back?
No idea.
Ask me in 18 more years,
Yeah, ask
right?
me in 18 years. I mean, I think fundamentally, you know, unless you kill every single cell associated with a tumor, you get regression. That's kind of fundamental. And to have this thing really effectively work, I think you have to combine kind of the anti-tumor effect with some sort of immune response, which bacteria are very useful for generating specific immune responses as well. But it would be a separate set of devices that you would have to build into these things that would elicit immune responses that result in prolonged regression.
So we're at that special point in the evening where we're going to talk about ethics.
Okay.
And I think this is really important for the public to consider from the perspective of there's been a lot of science that's been undertaken over the past, I would say 15 to 20 years without significant public input, whether it be genetic engineering or stem cell development, that has led to a lot of controversy in the public. And we're in a different situation with synthetic biology, where this is the infancy in many ways of the field. And right now they're soliciting a lot of input on the ethical and social challenges of moving this science forward. And I think this is the space, more so than anything else, where the public deserves and should put in their input. Because if it's not ethically and socially palatable to us, why invest? I'm guessing that the public funds most of your work.
Certainly, yes.
So do you agree with kind of the...
There are definitely ethical issues in here, and you know, it's something actually that's talked about almost as much as the science itself as it applies to genetic engineering. And I think part of this is people are a little scared of the technology because biology is foreign to people. If you're talking about baby boom generation people, the biology they learned in high school wasn't this stuff. Even when I was in high school, it was only 50-50. It was still, as we call it now, field and stream biology. It was learning about animals and plants. Whereas today in high school, maybe not in high school, but certainly in college, when you take basic biology, it is all about this molecular stuff. So it's less foreign, but you know, people don't get biology out there, and that scares people. So we have that issue. But also there are some real concerns in here, and the concerns kind of fall into two categories: biosafety and biosecurity. So biosecurity is the scenario where someone purposefully does something dangerous with genetic engineering that is really bad. So
Are we talking about a tomato that will never freeze, or bioterrorism?
We're talking about bioterrorism. That is the fear, that someone will make ricin E. coli in their closet, or someone will synthesize smallpox and put it in your water supply, something like that. And the focus of the concerns of biosecurity are principally on the DNA synthesis technology itself, not necessarily this kind of design process per se, because there's plenty of nastiness that you could make with the
pause you for one
technology.
second. Does that land as like bioterrorism, biosecurity as a legitimate concern here? Yeah. So I actually want to hear what they have to say first.
Well, we actually have already had something, but I don't think it is. Bioterrorism means that they're like non-governmental people. It's the labs. I mean, Monsanto is a bioterrorist, in my opinion, who is actually funding a lot of the malaria research.
And bioterrorism, from your perspective, why is that such an issue?
Well, it's one of a lot of issues. I think that somebody's going to do something and there's going to be an unexpected result. And that scares me.
All right. Oh, what about you, Al?
In a lab situation, they're going to do something and they're going to get a...
So it's not going to be like out of the movies where somebody is walking around Manhattan and Mel Gibson has to come save the day. It's something a little more, there's just an accident in the lab.
So this is more what people refer to as biosafety, where it's unintentional.
The Andromeda strain.
Yeah, and I mean there have been a lot of movies. So specifically with anti-cancer viruses, the recent was Will Smith movie. I forget what it was called. Where everybody turned into zombies.
But apparently people have created ricin in amateur environments, and that's what I'm more afraid of personally. It sounds like you have to work very hard to replicate these things. It's sort of a double-edged sword. On the one hand, you have to work very hard to replicate it, but you'd like to be able to replicate it better or get it self-replicating, but that's the dangerous thing.
Well, all these systems are fundamentally self-replicating, but you would have to intentionally make ricin to get ricin. You couldn't get ricin by accident. But there's the fear with the DNA synthesis technology that, some kid in their closet's going to think it's cool to make ricin. And then you have a security problem. So
let me ask you from this perspective. Is, in your mind, biosecurity a legitimate point of concern?
Oh, yeah.
And then how is the science community addressing that?
So right now, you fundamentally would have to have access to the DNA synthesis, which is exclusively present in companies. And when you ask these companies to synthesize a DNA, first of all, they ask you questions about it. So you have to tell them if it's a virulence factor. But also, they're going to do something called a blast search. A blast search, basically, you can take a DNA sequence and compare it to every known DNA sequence and see if it's at all similar. And they have a huge list of things that are no-nos. And they compare what you're asking to synthesize, they compare it to that list. And what they do, if you do that, I don't know. But I'm guessing they call the FBI. Or they just refuse to make it. I don't know what the response is. But that exists. That kind of, if you call that an administrative control.
That's self-imposed. The regulation is imposed by who?
I don't know if that is legally imposed or it's just the DNA synthesis companies have agreed to do that. I don't know if it's legally, it's certainly in the United States there's no law saying they have to do that. And largely this has been driven in the EU. They were the first to initiate this, but they all do it now.
We've had lots of conversations here about regulation. Shocking. Yeah. But it always seems to come back to the EU is leading the charge. The FDA not quite doing anything.
Well, part of it is, the new technology that scares people and why, what's really launched the new conversation about this is the DNA synthesis. That is a pretty new technology. It wasn't around in the 1970s when the first round of bioethics conversations were taking place. But a lot of the issues associated with environmental release and the regulatory framework for those was put into place in the 1970s. And there are fairly detailed rules associated with that. But those aren't rules about the composition of the organisms. They're more rules about, well, actually it is somewhat composition. There are specific rules about the use of virulence factors in organisms and the use of antibiotic markers in environmental release applications or large volume applications. So, first of all, it's not the case that there are no laws. I think some people don't realize that. But actually the laws were put into place in the 70s for the first round. And the question now is, what has to be changed? What is different now? And there are some things that are different in the way we're approaching things that you can't look at the problem of regulation the same way as when you were only really talking about putting one gene at a time into an organism.
When I was a freshman in college, one of my roommates was working on putting insulin into bacteria. Mm-hmm. And they couldn't do it. They weren't allowed to do it. The regulation said, oh, you're not allowed to do it. It went on, and we heard endless conversations about how dangerous this was to put insulin into E. coli. Well, as a physician, I give insulin every day that comes from E. coli, and we think of it as absolutely wonderful stuff, and I'm glad someone actually did that. And one of the things I would say about bioterrorism is there have been 3 bioterrorism attacks where people put ordinary bacteria into people's food. Ordinary bacteria can be highly toxic. You don't have to make special bioengineered
There's
bacteria.
no shortage of natural nastiness.
There's natural nastiness, and you don't really need to make them bioengineered to make them horrible. So we should really think about bacteria as dangerous things on their own, and you don't need to make one to be a terrorist.
Right. Yeah, I think that's a very important point. And the fact that those bad things have existed for a very long time means that regulators have had a very long time to think about how to control the access to and the penalties for playing with those organisms.
And we all know how to wash our hands and be careful about bacteria.
I'm sure we could go to a college dorm and find some nastiness right now. How is the scientific community involving the public in these kinds of discussions, though?
So the conversation on bioethics are not just scientists. In fact, I would say it tends to be more people like anthropologists and sociologists and philosophers who...
Because that's where I go when I'm looking for regulation. I don't know.
Well, you know, I mean, the average...
I don't know any
The average
anthropologists.
Joe isn't going to go to a conference to talk about bioethics, but certainly these conversations take place. Drew Endy was here in San Francisco at Fort Mason for a public conversation about it. And there are events like this that happen from time to time. There certainly are plenty of articles like the Science Friday type thing in mainstream media that discuss these issues. So it is out there, but I think also it's not on people's minds that closely that it pops its head up that clearly.
So I'll ask you a simple question. Should it?
I think whenever you're dealing with something this technical, most people are not going to understand it deeply enough to make the smart decisions about what the rules are. But then there's some things that are more global. Like, I mean, the stem cell thing was very mainstream. And the reason for it was it had nothing to do with biosafety or biosecurity. It was more of a moral sort of question. And when people are polled about the morality of genetic manipulation of various organisms, it's an interesting graph because the extreme majority of people don't care about what's done to bacteria. It's generally viewed as they're such low life forms that it's just not immoral to do anything to them. But when you start asking people, is it okay to genetically manipulate human beings, totally opposite response. I mean, it's like, no, don't do anything to a human. But when we're talking about synthetic biology, at least right now, we're principally talking about engineering bacteria and yeast. And the sort of applications we're talking about are these very chemical sort of applications that just don't stir that immorality fear that the manipulations of human beings get.
Do people out here have more social, ethical questions?
Yeah, as Kay was mentioning a moment ago about Monsanto and the corn engineering situation where this is corn that does not reproduce. And number two, if somebody, an adjacent farmer has a plot of land, gets the corn, plants it, Monsanto sues them out of business.
Right.
And it's like, that seems like a fairly heavy-duty moral situation that we have to consider. And right now, it looks like everything's in Monsanto's favor.
Yeah, so I think part of the reason genetic engineering has gotten a bad reputation has been that the big companies in it have been very aggressive with IP, intellectual property, and distribution of the products. So they'll put these things into their seeds that make it so you can't regrow them. You have to keep buying them from the company, which obviously makes them a lot of money, but makes them very unpopular with poor farmers. So there are a lot of ethics that just have to do with, it's nothing to do with the technology per se. It's how the technology is distributed. You know, when you're dealing with something like a food crop, or in the case of biofuels, I think similarly you're going to have this sort of thing.
Incidentally, do you know what the EU's position on this might be, since they seem to be the leaders in bioethics?
I have no idea in that sort of space of it. I mean, I think, you know, those are things that, you know, Microsoft has a bad rap too for being overly aggressive with their intellectual property and, you know, how they interact with other companies. But
I think Microsoft has a bad rap because my Vista machine just doesn't work.
Well, that's the one you rap. But, you know, I guess I'm dating myself now, but like 10 years ago, certainly the concern with Microsoft was their business practices, not the ethics of the system itself. And those issues are kind of, they're a separate category of ethical issues from the biosafety and biosecurity, but they're very important in this space because traditionally the companies have not been good citizens.
So there's a great report that was released by the UK late last year that lists out 5 areas of key ethical concerns for this synthetic biology. And the one that we haven't really addressed is kind of building upon his question, is rampant patenting of these sequences going to happen when in fact this is information that is essentially innately us.
Yeah.
Like you're patenting, goes one step beyond our food. This is patenting something a sequence that might exist in us naturally.
Absolutely. So this is, I would say, if you were to ask scientists about the ethical questions, what they're principally worried about, because it affects the field itself, is how people are doing patenting and intellectual property protection in the area of genetic engineering. Now, traditionally, since we were focusing on single genes, the product of your invention was a single gene in an organism. So there is a laundry list of putting a single gene into an organism to do something. And the problem with that is now when we say, okay, I want to put 10 genes into the organism, but it's going to be the same components that you guys were talking about before, but I'm talking about now an invention that's a higher order invention than the first one. My 10-gene organism is certainly a new invention and it is patentable in and of itself, but since there are patents already on the on all the 10 components of my system, I, in theory, have to get permission to sell that product to all 10 of those companies that have their product. And a lot of the companies that have patents on some of the more useful parts, one of the famous ones is zinc fingers, do have very aggressive policies towards controlling access to their sequences. So that is a major issue.
We have a question up front.
Would you say that there are any situations where there's too much concerns about ethics, or where it's going way overage, where people are worrying about things that they shouldn't? Like, for example, I read that there was a big freak out just at the beginning of genetic engineering, some sort of generalized "don't play God even if you save lives" thing. And I've read about a scientist who had created a drug that instantly cured drug addiction, but wasn't allowed to test it despite willing subjects begging him to give it to them.
Well, I mean, if people are concerned about something, then there is no ways to discuss in the ethics associated with it. But I think it is also true that there are some times when the debate around ethics gets off course, or is kind of looking over here at a problem when we're talking about something over here. When you're talking about the ethics of doing design of multi-gene systems, that's actually quite orthogonal to the technology that is a foundational technology of gene synthesis. And discussing, the right regulatory framework for gene synthesis is a very important discussion because it is dangerous. I mean, it's dangerous if you're not doing it in a safe manner. If you're making the sequences of things that are dangerous, it's going to be dangerous technology. But making GFP ain't going to hurt anybody. And there's this tendency to kind of talk about making smallpox when what the people are doing in the lab is playing around with melamine responsive GFPs. And it's clear that that space doesn't have any of the biosafety or biosecurity concerns that you have when you're talking about making a smallpox sequence. The problem is they're tied together because of the underlying foundational technology of gene synthesis that's needed to do either activity.
Are there any other ethics questions before we move on to the lightning round? Oh, there's plenty.
So on that topic of, back to the security issue and the underlying technology that doesn't have direct ethical implications, like it's not a smallpox sequence. But if you were to make discoveries or developments that made it easier for people made this technology more accessible, would you feel that you should go ahead and publish that and share that globally? And I bring this up thinking about things like RSA encryption and the United States export restrictions on that, and nuclear technology, and non-proliferation. Basically, it seems like a lot of times we feel like a technology could be used for dangerous things, so we try and either suppress it or limit its distribution.
Yeah.
On then the other side of that coin is that if you try and suppress it or limit it, you discovered it. Somebody else in some other country, maybe with different ideals than you, is probably going to develop it too. So how do you approach that problem?
I mean, it's an issue, and the main issue with it is the cat's already out of the bag on some of these technologies. I mean, you can't put the genie back in the bottle on gene synthesis, because it's, the synthesizer's been around since I think the 60s.
I think about the in vitro assembly process, and it's talking about using robotics.
Well, the robotics is all off the shelf as well. So, I mean, I think at some point what I think you'll probably see is that the hardware aspects of this, which will always be intensive, it's not like you're going to throw together that Velociraptor or whatever it was called robot in your garage. It's a fairly advanced piece of robotics. And that will probably be the control point for the administrative controls of the technology rather than the information about how to use it, simply because the information about how to use it would be really hard to contain. But you can easily watch the sales of that robot and find out who has it. And that's probably what will happen.
How confident are you that on a 10 to 20 year time frame, other countries, other smaller sects, or maybe not governments or large companies, but individuals that have scientific knowledge, experience, and funding will develop the same robotic technology in parallel, even though it is French? Give us an idea of how complex this is and how difficult that would be on a large enough time.
Or, as Fox News would put it, is North Korea going to develop the in vitro robot, or have they already?
They probably already have, but they recognize too that there are easier ways to get your hands on the biological agents than to synthesize them.
So that's a good argument. Is that there's easier ways to do biotech stuff. To do nasty stuff. So
I think that's the reality of it, is that they're all, if you're North Korea and you're a real country with, millions of people who get sick with pathogens, you can probably acquire any infectious agent you want just by the infections of your own population. And the motivation for synthesizing that natural agent rather than just isolating it from your backyard. I mean, if you're talking about anthrax, you can find that in your backyard. You can't turn it into a weaponizable agent without a lot of technology. Distribution of bioterror sort of things or bioweapons has always been the major hurdle for people to actually do something with them. But getting your hands on the biological organism itself, it's not clear that synthesis is even that useful of a technology for getting your hands on it. I think that's the reality of it, but the question is, how do you control it? And I think you have to track the components the same way you would track the components of a nuclear weapon sort of facility. And if you're talking about something like these picoraptor type things, there's a lot of parts that go into it, right? It's not just like a single device. It's a hierarchical device where at the core you have things like piezoelectric valves, and you could track piezoelectric valve technology just as much as you could track the robot.
So to kind of sum up this in a bottom line, keeping the technology from being something that someone can off the shelf build, that's not hard, no problem. And in terms of someone developing the entire technology from scratch, no more feasible than them building their own nuclear warhead.
Yeah, that's definitely the case, yeah.
All right, we have a few minutes left, so we're going to jump into the lightning round in a second, which is exactly as fun as it sounds. So, quick questions, quick answers.
Okay.
One quick addendum to the question before is Chris does do a lot of work with open source availability of his research and technology, and there are lots of pieces about it. Chief among them, like openwetware.org, does a really good job explaining how it facilitates expanding the availability of this technology and a lot of discoveries among collaborative science groups. So really interesting. I was going to write a little piece about it tomorrow. So if you're interested in that sort of open source availability of this technology, there's some great sources out there. All right, lightning round. Here we go.
Unintentional organism-to-organism or field-to-field transfer in the case of biofuel crops. Could you address?
So there's a big line that you need to be very conscious if you're stepping across it, which is creating an organism that actually can survive in a natural environment. So I think that that is fundamental in designing these organisms, that you engineer them such that they, first and foremost, you look for the applications where it is not a natural environment. So a bioreactor is not a natural environment. There are lots of things you could do to the organism to make it such that it can't survive in the soil, it can't survive in a pond, but you could make it such that it can survive in this very restricted environment. And I think, but how you go about safely doing things that involve environmental release, I think this is the issue we've had with the crops thus far, is that you're fundamentally making an organism that can survive in the environment, and you're crossing that line, and how do you safely cross that line is an open question.
Would you say that the control over the resources for biological engineering can be compared to the control over freedom of speech? Like, would you say that they're at all similar? Because freedom of speech everyone has, and there's really no way to completely control it. So would you say that it could be compared to that? Because bacteria is everywhere. Everyone has resources.
Are you asking that in a moral sense or just administrative sense?
Moral sense.
Because we could have laws where we put people into jail for things they say, but it's a human right. We don't do that. Now, do you have a human right to genetically engineer? That seems like a stretch to me, but.
Well, there have been problems caused because of people abusing freedom of speech. And we don't get rid of that, do we?
No, we don't.
It's just to do this work. We make them go through the pain of getting a doctorate and then becoming an assistant professor and begging for money. So that way it's a real
the control
dissuasion to
mechanisms
it.
thus far have been administrative controls. So basically there's a national agency like Public Health, PHA I think is what it's called, that kind of passes down the authorization, to give authorization to the states, which passes it down to the institutions, such that at the end of the day, I have to fill out a form saying what I'm doing to my UC Berkeley biosafety committee, and they look it over, and they have a bunch of criteria that they're looking for. And you know, if I say I'm gonna work with ricin, I better have a damn good reason for doing it, and I better have some really good containment facilities for working with it. And then, you know, if UC Berkeley signs off on it, and then the state signs off on UC Berkeley having done it, then it, you know, kind of passes its way up. So that is the way it works right now, is administrative controls. And then the way that works in practice, like where it stops you from going further with your ricin is, let's say I want to acquire the ricin, an organism that has ricin from one of the bodies that can sell to you, like a gene synthesis company or one of the biological kind of warehousing companies. They're going to ask you for your biosafety authorization number. You know, that's going to be some stamp of approval from UC Berkeley saying that you have permission to work with these agents. And if you can't give them that document, they're not going to send it to you. So that's how it works right now.
So we have one more, go for it.
Hello? The Biological Materials Liberation Front notwithstanding, are these genes currently being regulated like drugs? So do they have, the patents on them have the same, is it 7-year lifespan after which you could possibly have a generic?
You're talking about the patents?
Yes.
Yeah, the patents, like any other patent, they last 17 years and then they eclipse. And I think a lot of the real early stuff, like for example, T7 RNA polymerase is very commonly used for the expression of proteins. And traditionally, it's been under patent protection. And it doesn't really affect us academics because the companies don't care about us. But for industry people, they have to shell out a lot of money to use that system. But that stuff is all kind of coming off of patent about right now. So a lot of the real early stuff is kind of free again.
So you're saying that academics don't necessarily have to pay for these?
Legally, so it's a legal gray area. There is no true research exception to patents, but in practice, because we don't make any money, we don't have deep enough pockets for the companies that care about us.
Somewhere a patent lawyer is unhappy. I can feel it right now. All right, we're on to our last question of the evening.
My understanding of your work to hunt down cancerous tumors and kill them, do you have any effort on just strictly the early detection of cancer? And is this ultimately going to beat out the MRI and other kinds of imaging technologies?
I mean, I definitely don't work on that myself, and I'm not sure that anyone in this space works on that. I mean, you could, for example, engineer cells that were biosensors, that were going to look for specific epitopes or biomarkers and say turn green when they see them. So you would have a little crop of bacteria on a piece of paper or something in the clinic, and you'd spot some saliva or some blood on it and see if they turn green. And I think that is a real application out there that just hasn't been done yet. And you could also imagine having technologies that involve engineered cells for the discovery of new biomarkers. But in general, once you know what the biomarkers are associated with something, there are existing ways to develop a detection technology for the biomarkers. So the main problem with biomarkers is finding them, not so much detecting them once you know what to look for.
With that, we have nothing left to do but give Chris a big hand for coming out tonight. I am so convinced that to not do any of this work at home, I think you really, really had a big impact on me. We're here the third Monday of every month. We'll be back next month again. If you're interested in going on the Mission Science Workshops field trip to Mission High and plan like you're in science class again, just come see me. We'll talk about it. It is next Tuesday night. Other than that, thank you for coming. Big hand for Atlas Cafe for always doing a good job for us. And we'll see you again. Thanks.